Is this file visible to you?
Yeah, I can see it.
So this is the topic of nucleic acids.
Yeah,
And you have done it in bio as well.
yeah, I have.
So these are the two nucleic acids on your screen. Left hand side, it is RNA. It's a single stranded molecule.
Yeah.
And on right hand side, it is DNA. Left hand side, it is RNA. And on right hand side, it is DNA. I think you must have done all these formulae in bio as well, I believe. Have you?
What
The
sorry, say that
formulas
again?
like, say, adenine, guanine, cytosine.
Well, we've just learned the names. We haven't done like we didn't study like any of the
Any of the
formulas.
formulae.
Yeah.
Aayush, even in chemistry, at grade 12th level, these formulas are non-evaluative.
What do you mean by that?
That means they don't come under marks. It means students need not to remember.
Yeah, because I think during our test we get a data sheet and all of the like the main nitrogen bases, they're on the sheet. So if the question asks like,
it would be available in the question paper as well.
Yeah.
That's the meaning of the word non-evaluative. They don't come under evaluation.
Yeah. Oh, okay. Yeah.
This is unwritten law all over the world. Up to grade 12, these are non-evaluative. They don't say that it is not included in the syllabus. They say this is non-evaluative. Non-evaluative means students can go a little bit easily.
Yeah, that makes.
Okay, let us start. I'll be taking up a brief. As in bio, you have studied the particles present in the nucleus of the cells which are responsible for heredity are called as chromosomes. You know it very well.
Yeah.
Which are made up of, now it is about chromosomes, made up of proteins and another type of biomolecules called as nucleic acids.
Yeah.
Chromosomes are made up of two parts. One is the protein part, and other is the nucleic acids. And it is the nucleic acid which is practically responsible for heredity.
Yeah.
And that too, ultimately we'll be studying that DNA part.
Yeah.
So these derive the name from the fact that the first nucleic acid was obtained from nucleus of pus cells and was acidic in nature. Since it's a chemistry class, we must be very sure how much acidic. Very, very poor.
Oh, okay.
Very, very poor. These cannot turn blue litmus red. So this acidic nature is practically high, a very small amount as far as the other acids that we study in chemistry from the acidic point of view.
Yeah.
So this should be clear to you right on the onset that these are very, very poor acids if we go by the acidic nature.
Yeah.
These are mainly of two types, deoxyribonucleic acids and ribonucleic acids. DNA and RNA, very famous. Even a layman knows. DNA testing, DNA fingerprinting. At least DNA test, everybody knows. Even the man walking on the road knows DNA test. It is so popular. DNA is very common word. People almost tease each other when they want that this is in your DNA. You are like this because this is your DNA is like this. And all those people claiming and talking like this probably do not know what is DNA. The students from science background, they hardly talk like this sense. Otherwise, people from the literature or otherwise, they talk like this, that your DNA is very poor, your DNA is like this, that's why this happens. You must have heard, I believe.
Yeah, I have.
Okay, let's study. First of all, we come to the part of chemical composition of nucleic acids. As in proteins, we have studied chemical nature by hydrolysis. We keep on breaking proteins, and from proteins we get polypeptides, and from polypeptides we get amino acids finally. I have named only two other parts, polypeptides and proteins, the in-between one also. Now, if we go for hydrolysis of DNA or RNA, it yields three components. One is pentose sugar, then two types of heterocyclic nitrogen bases, purines and pyrimidines, along with phosphoric acid. Along with phosphoric acid. Okay. So one is pentose sugar. Now here comes the knowledge of carbohydrates. That would help you to understand that what is the structure of DNA. Pentose sugar. In RNA, there is ribose sugar, and in DNA, it is deoxyribose sugar. Now, coming to this part, we can show this organization like this, that nucleic acid is a polymer of nucleotides. Now, what is nucleotide? Combination of nucleoside and phosphate, two parts. Nucleosides are made up of further two parts, bases and sugar. Bases are purine and pyrimidines. The sugar is ribose and deoxyribose. This is just a memory chart, nothing else. Nothing else, okay? Now coming to the sugars. Now this is the sugar. If anybody looks at these formulas, just at first instance, he believes it's too much difficult. But things are rather simple. Now, how simple? Now, let's try to draw them. Little bit tied up?
Yeah, a little bit.
Enjoying weekends and enjoying holidays. It's difficult to have class. Okay, let's start. Just as you have written glucose, today now you write ribose. First of all, you write CHO. Since it is a pentose sugar, in total there will be 5 carbons. So now you write on the first carbon CHO group, and on the last carbon it is CH2OH. Is it okay? And if you remember, on the third carbon, the OH group is on left-hand side. It's the same way? Now write down the other ones. So it's very easy to write this. Now this is ribose sugar. This you can understand only by writing. Have you understood?
I understand.
Now what you do, you remove oxygen from second carbon. Then it becomes 2-deoxy. This is the meaning. I have removed oxygen from second carbon. The rest remains the same. HOH. CH2OH. Naturally, you can carry out its name as 2-deoxy. D means removal. Removal of what? Removal of oxygen. So 2-deoxy. Ribose sugar. Sugar is because of the sweetness. Saccharide. So got both the molecules, ribose and 2-deoxyribose.
Just one moment.
Okay. It's just a game for you now, I believe, Aayush. We have done glucose. It's on the parallel lines.
Yeah.
Now what you do, you make a ring of it. How? This pi bond shifts towards oxygen. Look at the screen. Pi bond shifts towards oxygen.
Yeah.
And then oxygen from the second last carbon. So this hydrogen from the second last carbon goes to O.
Yeah.
Just as in glucose. It's the same story. But in glucose, the ring was of 6 atoms.
Yeah.
Pyranose structure. Here it would be
so nice.
furanose, furan based. It goes like this.
Yeah.
Is it okay? Now you make its ring structure. How? So on the first carbon, you can write down CHOH. Then CHOH. COHH. CH. CH2OH. Like this. Is it okay?
Yeah.
We're just playing with it. Similarly, CHOH, HOH, OHH, we have to remove this oxygen, second one. Is it okay?
Yeah.
Now this is a 5-member ring. You understand? 4 members are carbon and 5th is oxygen.
Yeah.
And you know this structure. This is furan. It's a heterocyclic ring of 5 atoms in which 1 atom is oxygen.
Yeah.
And in glucose, you have studied this. Pyran.
Yeah.
This is furan.
Mm.
In ribose sugar and deoxyribose sugar, you have furan structure. What we say, it has furanose structure.
Mm.
Structure similar. You must have heard this word in bio, I believe, as well.
Yeah.
You must have heard this word.
I've heard the pyran for the glucose, but I haven't heard the furanose before. No.
This is okay, no issue. Furan is a 5-member ring.
Yeah.
It has furanose structure. And I believe now you can draw it comfortably. If I see, this is the structure. Now, how it is being drawn? Now, try to understand. Drying oxygen.
Yeah.
Like this. Okay. Now you start from the first atom. Like this. All the atoms on left-hand side above and the right-hand side below.
Yeah.
Okay. If we write as per the structure we have written, this H is on left-hand side above and OH below.
Yeah.
Is it okay? I'm rubbing a little bit of the molecules here so as to create some space. It is HOH, HH, OHH, and in the last it is H, and CH2OH.
Yeah.
Are you able to draw it on your own?
You said the left-hand side is
H,
on the top. Yeah.
OH below. You know that first atom, this first carbon, you can have OH on right-hand side as well as on left-hand side, as in glucose, alpha and beta forms.
Oh, okay, yeah.
So both possibilities exist. So I have taken one. So in second carbon, the numbering goes like this. It is first, it is two, it is third, it is fourth, and this is fifth. This is going to play a big role, this fifth carbon. This is going to join further.
Yeah.
So do you understand this structure?
Yeah, I understand it now.
Okay. You just need to understand. You need not to cram. The rest is similar. And it is being named as 1 dash, 2 dash, 3 dash, 4 dash, and 5 dash end in the numbering. This is a nomenclature that you use. You must have studied it in bio as well. 5 dash, 1 dash.
Yeah.
So these are the structures of glucose. This structures of ribose and deoxyribose, that means sugars. Now coming to the bases. There are two types of bases, purines and pyrimidines. I prefer that you write like this. Bases. Purines and pyrimidines. Now, purines are of two types. Adenine and guanine. Okay?
Yeah.
This is otherwise the basic purine structure. I have just marked it up. Need not to cram. They just as furanose, pyranose, similarly basic purine, basic pyrimidine. Okay?
Yeah.
Then pyrimidines are of three types. Cytosine, thiamine, and uracil. Adenine, guanine, cytosine, thiamine, and uracil. In the brackets, you write capital A for adenine, G for guanine, C for cytosine, T for thiamine, and U for uracil.
Yeah.
Is it okay?
Yeah.
In DNA, write down like this. Now, DNA contains adenine, guanine, cytosine, thiamine. And RNA contains adenine, guanine, cytosine, and uracil.
Yeah.
Thiamine is replaced by uracil. So both contain 4 nitrogen bases.
Yeah.
Then remember one thing more. Purine always pairs purine always pair with pyrimidine.
Yeah.
Revision of your bio class. And how they pair? Adenine pairs with thiamine by double hydrogen bond.
Yeah.
This is hydrogen bond, not a normal double bond. Adenine pairs with thiamine and guanine pairs with cytosine.
Yeah.
With triple hydrogen bond. In my student time, I used to memorize it like this at government college.
Oh, okay. Yeah.
This is at and this is government college, GC.
Yeah.
At government college, because then you never forget that which pairs with whom. Adenine with thiamine and guanine with cytosine. So these are the nitrogen bases.
Yeah.
Now this question figures in your exams. Nucleoside.
Yeah.
Write down with your own hands. Nucleosides. This base plus sugar is equal to nucleoside. Base plus sugar. It is nucleoside. And I have shown it, this combination. This is sugar and this is base.
Yeah.
And the bond between the two is called as glycosidic bond. Linkage with the sugar.
Oh, okay. Yeah.
The bond between nitrogen base, I have marked it like this, particularly marked it like this. Glycosidic bond. The bond linking nitrogen base with sugar. Wherever the word glyco comes, remember something related with the sugar.
Yeah.
The glycosidic bond. Why N? Because in nitrogen base, it's a nitrogen base.
Yeah.
And after their combination, their names changes. For example, the adenine becomes adenosine.
Yeah.
Guanine becomes guanosine. Cytosine becomes cytidine. Thymine becomes thymidine. And uracil becomes uridine. But nobody asks in the exams.
Yeah.
They ask, differentiate nucleoside and nucleotide. It's a normal standard question all over the world.
Yeah.
Now write down nucleotide. Nucleotide. It is base plus sugar plus phosphate. This combination we call it as nucleotide.
Yeah.
And then you remember like this. Memory tip. BS and BSP. Base and sugar. Base, sugar and phosphate.
Yeah.
The difference lies in the phosphate part. So this is nucleoside. And this is nucleotide.
Right. Yeah.
It means you can say that nucleoside is the building block of nucleotide.
Yeah.
Nucleoside ultimately builds up to nucleotide by combination of phosphate group.
Yeah.
This memory tips help. BS and then BSP. Is it okay?
Yeah.
And I have shown their structures like this. Nitrogen base, pentose sugar. This is nucleoside. And if you join even phosphate group, it becomes nucleotide.
Yeah.
And then their combinations like this. Now you understand this formulation. 1 dash 2 dash. Now this is deoxyribose sugar. You understand it now?
Yeah.
This is 3 dash, 4 dash, and 5 dash. You know the structure of phosphoric acid?
Yeah.
It is H3PO4. So what happens? Now OH part of sugar combined with H of phosphoric acid.
Yeah.
And the bond is formed. Got it?
Yeah.
So these three components together make one nucleotide. Nitrogen base, sugar, and phosphate.
Yeah.
This line is very important and figure in your examination, even it may figure in your bio exams. Nucleotides are joined together by phosphodiester bond or linkage. It means the same. Between 5 dash and 3 dash carbon atoms of pentose sugar. It's a technical point, very important point.
Yeah.
Two nucleotides joined together by which bond? The answer is phosphodiester bond.
Yeah.
Two nucleotides. And their joining point is 5 dash and 3 dash end.
Yeah.
Now here I have shown it, that how they join. This is 3 dash end of one side.
Yeah.
And this is 5 dash end of the other side.
Oh, okay. Yeah. That makes
And
sense.
this bond linking together is called as phosphodiester linkage.
Yeah. Yeah.
Is it clear to you?
Yeah.
Now on one side, I'm showing it like this. This is phosphoric acid. This is OH of one side and H of the other. The water is removed. You
Yeah.
get only O part here.
Mm.
You need not to draw this. You just have to make these things in your memory.
Yeah. Yeah.
What two nucleotides joined together by phosphodiester bond. Just as if you remember, peptide bond. What was the peptide bond? Linking two amino acids. Got it? You remember?
Yeah.
And we used to call that as dipeptide. Similarly, it is phosphodiester. Okay?
Yeah.
Three basic components of DNA are clear to you? Base.
What was it?
Three basic components of DNA. Base, sugar, and phosphate. Base and sugar join to form nucleoside. Base, sugar, and phosphate join to form nucleotide.
Yeah.
Base and sugar joined by glycosidic bond. And two nucleotides joined together by phosphodiester bond. It's all chemistry now. Then coming to structure of nucleic acids. Similarly, as in it was there in case of proteins, we can study its structure comfortably by dividing it into two components, primary and the secondary structure. And again, the meaning is also the same. Primary tells us the composition part, and secondary tells us the conformational part, that how it is being arranged.
Yeah.
So primary structure. Nucleic acids are polynucleotides, means formed by combination of thousands of molecules of nucleotides.
Yeah.
How those molecules would combine with elimination of water molecules. And the student of chemistry understands it very well. The condensation reaction always occurs with elimination of water molecule.
Yeah.
Now during polymerization, now see and focus, 5-CH2OH hydroxyl group of the sugar residue of one nucleotide combines with one of the OH group of the phosphoric acid. I have already explained to you.
Yeah.
Just as This is CH2OH combining with the OH part of the phosphoric acid.
Yeah.
At C3 at C-3 end. C-3 end is this much. This is C3.
Yeah.
And this is C5. You understand this difference?
Yeah.
Because numbering starts from this part. 1, 2, 3, 4, and 5. 1 dash, 2 dash, 3 dash, 4 dash, and 5 dash. Is it okay?
Yeah. Yeah.
Thus, the backbone of nucleic acids consists of alternating sugar phosphate residues. Each sugar on this backbone is further connected to 4 nitrogen bases. You know all this thing now. 4 nitrogen bases are attached to sugar phosphate backbone of nucleotide chain called as primary structure of nucleic acids. This is how they join together. This is adenine, thymine, guanine, cytosine, combining with sugar and phosphate. Sugar and phosphate, sugar and phosphate, sugar and phosphate. It is nucleotide.
Yeah.
And phosphate joins with sugar. This is phosphodiester bond, combining 2 nucleotides. Otherwise, all nucleotides are single in themselves. Got it?
Yeah.
This is the simple way to show it. 3 dash end of one side combined with 5 dash end of the other side.
Yeah.
This is 1 nucleotide, 2nd nucleotide, 3rd nucleotide, and so on. For example, this is tetranucleotide because I have shown it 4 nucleotides here. A, C, G, T.
Yeah.
Tetranucleotide. In your exam, from your examination angle, this is very important. Chergaff rule. Have you studied it in bio? I do believe you must have. This is going to figure in your bio exam as well, my dear.
I don't know. I've never heard of the name, but I might have been taught the rule.
It is too much famous with its name as well.
Yeah, I think I should have. Yeah.
As per the present day notation, it should be called as Kergef.
Okay.
Because CH sounds for K.
Yeah.
This has been decided by the IUPAC in the year 1997.
Oh, okay.
That you sound CH with the K sound. Because we call it as chemistry and not chemistry. We call chiral, not chiral or chiral.
Yeah.
It has been decided once for all. Because there was a problem in chemistry. And in India, we call it a Chopra-Kopra problem. You must have heard this surname, I think. Chopra.
Yeah.
This is Chopra and Kopra. Chopra-Kopra problem. This is CH, chiral or chiral. It is chiral. It has been decided by IUPAC in the year 1997. This is Chergaff rule. It simply says number of purines must be equal to number of pyrimidines. That's all.
Oh, okay.
Because they have to link together.
Yeah.
So in DNA molecule, as far as their base composition is concerned, total amount of purines must be equal to total amount of pyrimidines. That's all.
Yeah.
A plus G must be equal to C plus T. Because A is always going to pair with T.
Yeah.
And G with C.
Yeah.
If there are 100 A's, there must be 100 T's. If there are 1000 G's, then 1000 C's must be there. Because guanine is going to pair with cytosine.
Yeah.
At a government college. Now that tip helps. And you never make a mistake. So you remember only this formula, write down with your own hands. A plus G is equal to C plus T. But this is Chergaff rule. Kergaf rule. But in bio, or rather majority of people pronounce it as Chergaff. Is it okay?
Yeah.
So knowingly, I have made this as a balance, physical balance. Adenine must be equal to thymine.
Yeah.
And cytosine must be equal to guanine.
Yeah.
This 80 by GC ratio is very typical for the particular animal. It is this, for example, it is 1.52 in man.
Yeah.
In this 80 by GC ratio. And in Escherichia coli, it is 0.93. You studied in bio, I believe, Escherichia coli, E. coli.
Yeah. Yeah. Bacteria.
It is 80 by GC ratio.
Yeah.
If it is 1.53, what did you get from this? Which pairs would be more?
The 80.
80 pair.
Yeah.
And if it is 0.93, then GC pairs.
Yeah.
It means in human beings, 80 pairs are more than GC pairs. So simple to conclude. Now here comes the maths, not the bio. That in human beings, now in every bio book it is written that 80 by GC ratio is 1.52 in man. But many times students are not able to correlate that 80 pairs are more than GC pairs.
Yeah.
Got it?
Yeah.
So this is the ratio in different cases. Virus, bacteria, fungi, and higher eukaryote, human beings.
Yeah.
And there is one numerical problem because now the chemistry class. Very simple. 80 by GC ratio in E. coli is 0.93.
Yeah.
If the number of moles of adenine in its DNA sample are 4,65,000. I think in UK you must be calling it like this. 465,000?
Yeah, 465,000.
465,000.
Yeah.
You don't talk in terms of lakhs, I believe. That's
No.
in India only. 465,000. It's okay?
Yeah.
465,000. Calculate the number of moles of guanine.
Okay.
It is 80 by GC. If the number of moles of adenine is 465,000,
yeah,
then you have to calculate number of moles of guanine. Can you? So that the ratio is 0.93. If 465,000 adenines are there, similarly, so many thymines must have been there.
Yeah.
And to get that ratio, it means there must be 500,000 guanine and 500,000 cytosine.
Yeah.
The answer is 500,000. That's all.
Yeah.
Very simple numerical.
Yeah, it's a lot easier now that I understand it. Because the like for biology, these are the like difficulty of calculations. It's not going to go like that much difficult. It's just understanding it that gets you confused.
I know. Here comes the secondary structure of DNA, double helix structure. This you must have studied.
Yeah.
Proposed by Watson and Crick. This is the model you are looking in the image. This is double helical structure.
Wait, so does RNA only exist as a primary structure then?
No, RNA also exists in secondary structure.
Also, but how would it have
it goes like this, that's all. Single-stranded. Just like a ribbon coiling on its own.
Yeah.
And these are like stairs going round up. You must have seen such stairs, I think.
Yeah. No, yeah, I've seen the different types of the RNA and everything, but I thought like you can only have like say equal adenine and thymine only if there are two strands, because the A bonds to the T.
Oh, that is
C.
okay. That is okay. They are going to be equal in that, but even in RNA, they get bonded.
Okay.
Why not? This is the point from where we started.
Hmm.
Now you can see the strands recoil among each other.
Okay.
It's a single-stranded molecule.
Yeah.
And in biology, this RNA is very important for protein synthesis, for transcription.
Hmm.
We won't be going into those details in chemistry class.
Yeah, that's fine.
Is it okay?
Yeah,
So far?
yeah, it's fine. Yeah.
It's 80 by GC ratio. The simplest way to draw it, I think you must have studied it in bio. Draw like this with your own hands. The simplest way. This is adenine.
Hmm.
Joining with thymine. Guanine joining with cytosine. Cytosine joining with guanine. Thymine joining with adenine.
Hmm.
Is it okay?
Yeah.
On one side, you draw this pentose sugar like this. The simplest way to write pentose sugar. You understand this?
Hmm.
It cannot be further simplified.
Yeah.
Pentose sugar. We are drawing a cyclopentane and then putting S over here.
Yeah.
Pentose sugar. Pentose sugar. Pentose sugar. And on the other side, you draw in the opposite manner.
Yeah.
Because two strands run in opposite direction.
Yeah. Anti-parallel.
It's a revision of bio class.
Hmm.
This is pentose sugar. But the structures become more clear in chemistry class, the chemical formula. Now, are you able to draw it?
Yeah.
Then join them with common phosphorus. Like this. Is it okay?
Hmm.
Similarly, on this side. Like this. Do you get it?
Yeah, I understand.
And I think that you can draw it very comfortably now.
Hmm.
This is double-stranded.
Yeah.
And to make you further clear, and just to put it in your memory, this is thymine, and this is adenine, and joined by hydrogen bonds.
Yeah.
2 hydrogen bonds. Similarly, this is guanine and cytosine combination. So 3 hydrogen bonds.
Yeah.
Got it?
Yeah.
And this is how 2 DNA strands run in opposite manner. I hope you can draw it very comfortably.
Yeah.
Okay. The diameter of double helix is 2 nanometer. Because these are factual questions. Double helical structure repeats itself at interval of 3.4 nanometers. That means one complete turn is of 3.4 nanometer.
Oh, okay. Yeah.
which corresponds to 10 base pairs.
Mm.
Now it becomes now bio class, just factual things.
Yeah.
In bio, you remember all this. It's
Mm.
memory base now. Two kinds of grooves, one major and one minor, is evident in the structure. And I have just opened it up like a normal stair,
Mm.
double-stranded. The major groove, the bigger turn, the minor groove, the smaller turn. You need not to remember, I believe.
Yeah.
But one thing is sure, I think that you must have memorized by now. Thymine pairing with adenine and cytosine pairing with guanine need not to do more than this.
Mm.
Then there is a conformation, B conformation of DNA having right-handed helix is the most stable one. These are the details. BDNA and ZDNA.
Mm.
Have you gone up to this level in bio or not?
No, I've never heard of this being said DNA.
That how it goes, you see, is focus on the blue color.
Yeah.
It is going like this. And on this side, it is going in the opposite side.
Yeah.
Okay? As per the strands.
Mm.
These have been given names on the basis of how the strands are going up.
Mm.
Now you understand, in B conformation, DNA having right-handed helices is the most stable. This is right-handed. And they keep on changing into one another.
Yeah.
And it's a diagram that we were drawing earlier on. This we are drawing?
Yeah. Yeah.
The temperature at which two strands separate completely is known as melting temperature.
Mm.
This is how two strands run.
Yeah.
And if they separate,
So,
the temperature at which they separate is called as melting temperature.
mm.
If I say that melting temperature of one of the person is higher, for example, this is a numerical given here. Two samples of DNA, A and B.
Yeah.
There is one museum known by the name Field Museum.
Mm.
This is in Chicago. One of the biggest museum in the world.
Mm.
Luckily, I have got opportunity to visit this.
Oh, okay, that's nice.
Aayush, they have one lab there where they are keeping DNA of all the species.
Oh, okay. Yeah.
It's the biggest DNA bank in the world.
Yeah.
The species that have occurred so far on this planet.
Mm.
They are just collecting all the data and putting in that bank for further study for the future generations to come.
Yeah.
Two samples of DNA, A and B, have melting temperatures 340 and 350. Can you draw a conclusion from this data regarding the base content? So normally we take it as a B sample of DNA, not Z.
Yeah. So would that just depend on the ratio of the
Yes,
ATCG?
yes. If temperature
Yeah.
is higher, do you get any idea whether AT pairs would be more or GC pairs will be more?
Would the GC pairs be more because
Yes,
it's more
GC pairs will
hydrogen
be
bonds?
more because they have a triple hydrogen bond.
Yeah, exactly.
Such a simple question and just pure application.
Yeah.
Okay. Now coming to secondary structure of RNA that you were asking.
Yeah.
Only single stranded. Actually, you don't find such images in the normal books.
Mm.
You don't find single stranded R, particularly the RNA part.
Yeah.
This is a base pair, how they are linked together. Now you can make a comparison. Now it is single stranded. and this is double-stranded. This is DNA and this is RNA.
Yeah.
The only difference is that thymine is being replaced by uracil.
Yeah. And RNA, isn't it like RNA only exists from 5' to 3', like the direction?
You can say it is halfway of DNA, that's all. Very simple way to memorize.
Yeah.
But there is one strand. It's hanging in the air, as believed, in comparison to stairs.
Yeah.
Because while studying or talking of DNA, stairs automatically run into our mind.
Yeah. They
The
always give that
simplest way.
example.
Because the reason being that right from your childhood, the day you started studying DNA, the example of stairs is given to you.
Yeah.
Then somehow we're not able to comprehend this RNA. It's a single-stranded, because since you have studied alpha helical structure of protein, you remember like this?
Yeah.
Single coiling. So that is possible because then hydrogen bonding takes place. You remember?
Yeah.
It is similar to that. Otherwise, nobody asks this structure of RNA.
Yeah.
This question, I believe, you can very easily answer. Difference between DNA and RNA, but you get it in your examinations.
Yeah.
DNA occurs in the nucleus, RNA in the cytoplasm. DNA contains deoxyribose sugar and RNA contains ribose sugars.
Yeah.
Here is thymine, and in RNA it is uracil.
Yeah.
It's double helical, it is single helical. This is the most important point. Genetic material, not a genetic material.
Yeah.
DNA can replicate itself. Replication means producing a copy of its own.
Yeah.
RNA cannot replicate. 6 differences are much more than sufficient for any question. And this you can remember effortlessly.
Yeah.
DNA is in the nucleus, RNA is in the cytoplasm. Then focus on the sugar part. Ribose in RNA and deoxyribose in DNA. Thymine in DNA and uracil in RNA. DNA is genetic material, RNA is not.
Yeah.
DNA is double-stranded, RNA is single-stranded. DNA can produce a copy of its own, and RNA cannot. So these are the 2 important processes. These are the 3. Replication, transcription, and translation.
Yeah. Those are very big questions in biology.
Very, very big questions. But you were doing it in bio, I think.
Yeah.
I would be focusing only on the replication part. Transcription and translation involves pure bio.
Yeah.
I won't be going into details at grade 12th level. I deal with these things at higher classes level. Transcription and translation. But since by the time student reaches those levels, they have done in bio. So things are not that difficult. We need not to repeat it in chemistry class. This word is very important, central dogma. These concepts constitute the central dogma of the molecular biology and were summarized by Crick in the following diagram. This is central dogma. DNA undergoes replication, then transcription and reverse transcription changes to RNA, then there is transition to protein. That's a translation part ultimately. Particular coding goes for particular protein. This DNA, transcription to RNA, then translation to protein. This is called as central dogma. You must have heard this word in bio, I think.
Yeah.
Central dogma. Replication is very simple. These 3-letter codon triplet, 20 different types of amino acids written in the language of 20 letters, but the hereditary message is written in the language of only 4 letters. It is written in code with each word of 3 letters, standing for a particular amino acid. You have studied this in bio?
Yeah.
In replication, let's focus on the first part, replication. This is how 2 strands of DNA open up. and the bonds between them get broken.
Yeah.
This you understand, I think.
Yeah.
Adenine, thymine, guanine, cytosine, and on this side there must be thymine, adenine, cytosine, guanine. This builds a strand of its own, and this builds a strand of its own.
Yeah.
Then here comes thymine. What will come here?
Adenine.
Adenine.
Cytosine. Go on.
Such a simple thing. This remains with the parents, and this goes to offsprings.
Yeah.
This is how this information is transferred. It is adenine, thymine, guanine, cytosine. Is it okay?
Yeah.
If you see these diagrams, and if you try to memorize, things will be difficult.
Yeah.
And if you apply your own brain and make your own base sequence, it is very, very comfortable.
Yeah.
This is the question that you get in exams. That one strand of DNA is given to you, make the other one.
Oh, okay.
Such a simple thing. On adenine, you put thymine.
Yeah.
On guanine, you put cytosine. On cytosine, you put guanine.
Guanine.
On adenine, you put thymine.
Yeah.
On adenine, you put thymine. On thymine, you put adenine.
Yeah.
Ultimately, where it goes? At government college, that's all.
Yeah.
That's a simple thing. If it comes like this, AGT, ACT, GAT, write the corresponding base sequence. What it will be?
TCA.
AGT would be equal to
TCA.
TCA. Such a simple thing. We should not spend more time on it. In bio class, these appear to be very difficult questions otherwise. It's very simple.
Yeah.
This is the question I've written in the exam. That's all. I won't be focusing on protein synthesis that you do in your bio class.
Yeah, that's fine. Yeah.
And that finishes this topic.
Yeah.
Anything else you want to ask on this topic?
From this topic, no. I understand it. Yeah, I understand everything well.
Okay. Should I stop recording for the moment?